Have you ever, like, stopped to think about what's going on inside you even when you're just sitting still? Like, all the stuff happening inside your cells.
Oh, yeah.
They're always working, right? And just like us, they can get old,
Yeah.
damaged, you know?
Mm-hmm.
And we usually think about, like, injuries when we think about cell death.
Right.
But there's this whole other world of cell life
Yeah.
and death
A hidden universe.
that's happening all the time, so.
Yeah, and when we talk about cell death, it's not just like one thing.
Right.
You know, it's a whole spectrum of processes.
Exactly.
Some very orderly, some less so,
Yeah.
but all crucial in their own way.
That's exactly what we're diving deep into today.
Yeah.
Like, forgetting the basic idea of
Mhm.
cells just ceasing to exist.
Right.
You, our listeners, shared some really cool material
Absolutely.
that opened my eyes to, like, the many different ways that
Yeah.
a cell can actually end its life.
It's really fascinating.
And it's way more complex and vital than I think we probably ever imagined.
Yeah, for sure. We've got some great sources to guide us today.
Yeah.
A review article titled Cell Death, a review of the major forms of apoptosis, necrosis, and autophagy, which lays out like the whole landscape for us.
Perfect.
And then we have a pathology lecture on cell death, offering a really cool clinical perspective.
Awesome.
And finally, another big review, Diversity and Complexity of Cell Death, a historical review.
Love it.
And this is so cool because it really shows how, like, our understanding of this whole thing has changed over time.
Yeah, totally.
Yeah.
So our mission for you today
Yeah.
is to unpack these different ways cells can die
Okay.
and understand not just the how, like the mechanics,
Mhm.
but the why these different processes matter for your health.
Right.
We're aiming for those aha moments,
Absolutely.
those surprising facts
Mhm.
that might just shift how you think about your own body
Yeah.
without, like, getting bogged down in a ton of technical details.
Right.
All right, let's unpack this, starting with what we considered initially like the two main forms of cell death.
Right. For a long time, we focused on these two distinct ways a cell could die. Necrosis and apoptosis.
Okay.
Let's start by looking at necrosis.
Necrosis. Okay, so the sources tell us that historically
Mhm.
this was seen as a pretty messy way,
Yeah.
uncontrolled way for a cell to die, usually caused by some injury,
Right.
like a trauma or an infection.
Yeah.
Sounds pretty violent.
Well, that's a good way to put it. The term itself, necrosis, it has roots in Greek.
Okay.
Necros means dead or a corpse.
Oh, wow.
And esphosis refers to a process.
Oh,
So in this case, like a process of destruction, degeneration.
process of death.
Yeah, and what's interesting is when this term first started appearing in the late 18th century,
Okay.
it was often used to describe the death of tissues
Mm-hmm.
from things like infection, inflammation,
Oh, interesting.
not necessarily the death of individual cells.
Oh, so who was it that really brought the focus down to what was happening at the cellular level?
So that key figure was Rudolf Virchow. Back in the mid-19th century,
Got it.
he really defined necrosis at the cellular level.
Okay.
He observed that in necrosis, the mortified cell is left in its external form.
Interesting.
And he contrasted this with what he called necrobiosis
Okay.
or shrinkage necrosis,
Okay.
where the cell kind of just disappears.
Oh, wow.
Later on, we started to see this idea of coagulative necrosis
Okay.
as like a specific pattern within this broader category.
Okay, so then what actually happens inside a cell when it undergoes necrosis? Like, what are the key things that we see?
Well, the most fundamental thing is the loss of the cell's structural integrity.
Right.
The internal components start to break down, and
Mhm.
the cell's contents
Uh-huh.
are released into the surrounding extracellular space.
Oh, so it's like spilling its guts.
Exactly, and this spilling out is why necrosis often triggers inflammation
Makes sense.
and can lead to more damage in the tissues around the dying cell.
Also, it's not just the cell itself dying, but it's affecting the surrounding tissue as well.
Right, it's not a clean and tidy exit.
What does this actually look like under a microscope?
So morphologically, we see some pretty distinct changes. The cell tends to swell up. Those little powerhouses of the cell, the mitochondria, become damaged.
Mhm.
The chromatin, which is the DNA, starts to clump together.
Uh-huh.
Then the nucleus itself might shrink, a stage we call pinosis,
Pinenosis.
followed by dissolving, which is karyolysis,
Derailed.
and finally breaking apart into fragments, karyorrhexis.
Wow, okay.
And something quite telling is the cytoplasm, the main body of the cell,
Yeah.
loses its ability to take up stains
Oh, interesting.
that we use in the lab to visualize cells.
No, it's like it loses its identity almost.
Yeah, it's really a meltdown.
Cellular meltdown. And the sources really emphasize that necrosis is always pathological, right? It's never
Yeah.
something that the body intentionally does in a healthy way.
Precisely.
Yeah.
It's always associated with some kind of cellular stress or injury that overwhelms the cell's ability to cope. And this leads to a sharp drop in the cell's energy production.
Okay.
ATP levels plummet.
Yeah.
The outer boundary of the cell, the membrane, loses its integrity, and the cell's machinery for making proteins and DNA just grinds to a halt.
Oh, wow. It really shuts down.
Yeah, it's a catastrophe.
So then what are some of the common things that can cause this kind of cellular disaster?
Oh, a really wide range of insults can lead to necrosis.
Okay.
So physical agents like extreme heat, think burns,
Yeah.
or extreme cold,
Got it.
like frostbite,
Okay.
chemical agents like mercury chloride,
Okay.
formaldehyde,
Yeah.
or even like everyday substances
Wow.
if they're in high enough concentrations, like strong alkalis.
Okay.
And then, as we said before, a lack of oxygen,
Yeah.
whether it's completely absent, anoxia,
Hypoxia.
a severe reduction, hypoxia,
Smirks.
or a restriction of blood flow, ischemia.
Ischemia.
These are all major triggers for necrosis.
So basically anything that like really stresses the cell out too much.
Yeah, exactly.
And then I noticed in the material, there were different types of necrosis mentioned.
Right.
So it's not just one way for this messy cell death to happen.
That's a very important point.
Okay.
The way necrosis looks under a microscope, the morphological appearance
Yeah.
can vary a lot depending on the tissue affected and the underlying cause.
Okay.
For instance, in coagulative necrosis,
Coagulative necrosis.
which is often seen when blood flow is blocked, like in heart attacks, myocardial infarct,
Myocardial infarct.
kidney infarcts, spleen infarcts.
Got it.
The proteins in the cell basically denature. They become misfolded, and the tissue becomes firm and pale.
Interesting.
Microscopically, it looks homogeneous,
Yeah.
acidophilic.
Okay.
The nucleus is lost, but the overall structure of the tissue is still recognizable.
Oh, so you can still see the outlines of the cells, even though they're dead.
It's like the ghost of the tissue remains.
Yeah, that's a good way to put it.
Yeah.
What about liquefactive necrosis?
Liquefactive
It
necrosis.
sounds liquidy.
It is, literally.
I remember
This
that.
type of necrosis involves the enzymatic digestion of the tissues.
Okay.
It's often associated with bacterial infections where you get pus.
Right.
That's the accumulation of neutrophils,
Okay.
a type of immune cell,
Got it.
and other inflammatory cells,
Okay.
along with the liquefied dead tissue.
Oh, so it's like a soup of
Yep.
immune cells and
Yeah.
dead tissue.
Brain tissue, which has a lot of lipids, also commonly undergoes liquefactive necrosis
Oh, interesting.
when it's damaged.
Okay.
Abscesses,
Mm-hmm.
which are those localized collections of pus,
Yeah.
and some malignant tumors can also show this.
Okay, so coagulative is firm, liquefactive is liquid, and goopy, what's next?
Fat necrosis.
Fat necrosis.
Yeah. Does that involve fat cells?
It sounds like it does.
It does. It specifically affects adipose tissue, which is our fat tissue.
Got it.
It's often seen in cases of acute pancreatitis,
Okay.
where these digestive enzymes, lipases, are released from the pancreas, and they start to break down the fat cells.
Okay.
The fatty acids that are produced then bind with calcium ions, forming these chalky, whitish deposits called calcium soaps.
Calcium soaps.
These are quite distinctive under a microscope
Yeah.
and can even be seen by the naked eye during surgery.
Oh, wow. So it's like the body's creating its own soap.
Right, in a pretty unfortunate situation.
Exactly.
Yeah.
Okay, the list goes on. Caseous necrosis.
Caseous necrosis.
The name makes me think of cheese.
That's a really good description.
Okay.
Macroscopically, caseous necrosis has this like amorphous cheese-like appearance.
Okay.
Whitish, soft, crumbly. It's classically found in infections like tuberculosis
Tuberculosis.
and sometimes in certain infarcts or malignancies.
Okay.
Microscopically, it looks granular
Okay.
and amorphous, meaning it lacks any clear structure, complete loss of any recognizable cellular detail.
So it's just like disintegrated completely.
Complete
Okay, then there's fibrinoid necrosis.
fibrinoid necrosis.
That sounds a little more specific in terms of what it affects.
It is.
Okay.
This type primarily affects the walls of blood vessels and connective tissue.
Okay.
The normal structure is replaced by this intensely acidophilic hyaline material that resembles fibrin,
Okay.
which is a protein involved in blood clotting.
Got it.
It's a hallmark of certain autoimmune diseases,
Okay.
sometimes called collagen diseases.
Interesting.
like rheumatic fever and rheumatoid arthritis.
Got it.
It can also be seen in atherosclerosis and peptic ulcers.
Okay, so we're really getting a tour of all the different pathological landscapes here.
We are?
What about hemorrhagic necrosis?
Hemorrhagic necrosis.
That sounds pretty straightforward.
Yeah, it is. It's simply necrosis with bleeding into the tissue.
Okay.
This is often a result of an obstruction of venous blood flow, which causes like a backup of blood, and then leakage into the tissues where the cells are also dying.
Right, so the blood can't get out.
Right, and the presence of blood can complicate the body's ability to clear away the dead tissue.
Makes sense.
Yeah.
And finally, the dramatic-sounding gangrene.
Gangrene?
I think most people have heard of that.
Right. Gangrene is basically necrosis with external factors superimposed on it.
Okay.
So there are a few main types.
Okay.
Dry gangrene is typically the result of ischemic coagulative necrosis of the extremities.
Like fingers and toes.
Right. Fingers or toes.
Okay.
The affected tissue becomes dark, dry, and shriveled
Okay.
due to the changes in hemoglobin and the loss of water.
There.
Wet gangrene, on the other hand, involves decomposition of the necrotic tissue by saprophytic and anaerobic bacteria.
Oh, so now we have bacteria involved.
Right, and this leads to putrefaction, a foul odor, and a softening of the tissue.
Right.
And then there's gas gangrene,
Gas king.
which is particularly severe and rapidly spreading.
Oh, wow.
It's caused by infection with Clostridium bacteria in wounds.
These bacteria release toxins that cause muscle death or myonecrosis
Myonecrosis.
and produce gas, hence the name.
Wow, that is quite a catalog of cellular destruction.
It is.
It really paints a clear picture of just how damaging uncontrolled cell death can be.
Yeah.
Now let's switch gears completely and talk about the other classic form of cell death, apoptosis.
Okay.
Now this is described as a much more organized and controlled affair.
Absolutely.
Okay.
Apoptosis is characterized by this programmed self-destructive process that occurs in response to very specific signals.
Okay.
So the term apoptosis itself was coined by Kerr, Wiley, and Curry in 1972.
Wow.
It comes from Greek. It means falling off,
Interesting.
like leaves from a tree in autumn.
That's kind of beautiful.
It is a beautiful way to think about it
Yeah.
because it suggests this beneficial loss without harm to the overall organism.
That's a much more poetic image than the cellular meltdown we talked about before.
But the sources also point out that the idea of this programmed cell death wasn't entirely new in 1972.
Right, that's a good point.
Yeah.
The concept of physiological or naturally occurring cell death was first presented by Carl Vogt way back in 1842.
Wow.
Later, in the late 19th century, Walther Fleming, who's also known for his work on chromosomes, observed a cellular process he called chromatolysis.
Chromatolysis.
During the degeneration of ovarian follicles, which we now understand to be related to apoptosis.
Oh, interesting.
The key difference with apoptosis, as we understand it today, is that it's a genetically controlled process.
Right.
It involves the activation of specific genes,
Yeah.
and it often involves breaking down the cell's DNA in a controlled way.
So it's not just random.
No, it's very organized.
Yeah.
And it can be triggered by both normal physiological signals and non-physiological or harmful stimuli.
So unlike necrosis, which is usually a response to injury, apoptosis isn't always a bad thing for the body.
It's not.
Okay.
Apoptosis plays these absolutely crucial roles in normal physiology.
Okay.
Think about embryogenesis.
Embryogenesis.
The development of an embryo.
Right.
Apoptosis helps to sculpt tissues and organs.
Oh, wow.
It gets rid of cells that are no longer needed.
So it's like cleaning up as it goes.
Yeah, it's involved in morphogenesis,
Morphogenesis.
the overall shaping of the body.
Oh,
And in processes like metamorphosis.
like when a tadpole turns into a frog.
Exactly, and loses its tail.
Yeah.
It's also essential for the regular turnover of cells in our tissues.
Oh.
The atrophy of tissues when they're no longer needed, like due to a loss of growth factors or hormones.
Right.
The involution of organs after they've served their purpose.
Like the uterus after childbirth.
Exactly.
Really?
And even in the regression of tumors
Oh, wow.
in some cases.
So it's almost like a cellular self-pruning mechanism that's essential for development and maintenance.
Precisely.
Okay.
So it helps control the density of cell populations. It removes cells that have been damaged by things like toxins or radiation.
Right, yeah.
And might become dangerous.
Yeah.
And as I mentioned, it's critical in the formation of organs,
Okay.
especially in systems like our skin and our immune system.
So many different roles.
Yeah, just to give you an idea of the scale of this.
Okay.
It's estimated that about 1 billion neutrophils,
Wow.
which are a type of white blood cell,
Okay.
undergo apoptosis every single day in a healthy adult male.
1 billion cells a day undergoing this controlled dismantling. That's wild.
It is.
So then what does this process actually look like at the cellular level? What are the key morphological hallmarks of apoptosis?
So unlike the swelling and bursting we see in necrosis,
Right.
apoptosis is characterized by cell shrinkage.
Okay.
A key distinguishing feature is chromatin condensation.
Chromatin condensation.
The DNA inside the nucleus packs together.
Okay.
The DNA is then fragmented by these enzymes called endonucleases,
Endonucleases.
which cut it into these fragments of roughly 200 base pairs in length.
Okay.
Then the plasma membrane, the outer boundary of the cell,
Mm-hmm.
starts to bleb, forming these little bubble-like protrusions
Okay.
called apoptotic bodies
With polygotic bodies.
that contain the cell's internal contents.
Mm-hmm.
These apoptotic bodies are then engulfed and digested by phagocytes,
Okay.
like macrophages, or even by neighboring cells.
Interesting.
This whole process happens without any leakage of the cell's internal enzymes
Okay.
into the surrounding tissue.
So no collateral damage.
Exactly, and crucially, without triggering inflammation.
Okay.
You also see the condensation and fragmentation of the nucleus,
Mm-hmm.
and in tissues like the linings of organs, the connections between cells,
Okay.
called desmosomes.
Desmosomes.
Got it.
Yeah. So it's a very contained and clean process
It is.
designed to avoid that collateral damage.
Exactly.
Now, the material mentioned two main pathways that can lead to apoptosis, the intrinsic and the extrinsic pathways.
Right.
Can you break those down for us?
Certainly the intrinsic pathway,
Okay.
also called the mitochondrial pathway,
Mitochondrial path.
is triggered by signals that originate within the cell itself.
From the inside.
Yeah, these signals are often a response to intracellular damage sensors detecting things,
Okay.
like low oxygen levels, hypoxia, presence of toxins, exposure to radiation,
Yeah.
high levels of reactive oxygen species,
Reactive oxygen species.
which can damage cellular components,
Right.
viral infections,
Okay.
or even the absence of survival signals,
Like,
cytokines, hormones, and growth factors.
got it.
This pathway involves the release of certain signaling molecules from the mitochondria, which are the cell's powerhouses,
Yeah.
and the activation of a family of enzymes called caspases,
Caspases.
specifically initiator caspases,
Okay.
like caspase 9 get activated,
Yep.
and they then go on to activate the executioner caspases,
Wow.
typically caspases 3, 6, and 7, which are the enzymes that actually carry out the dismantling of the cell.
Wow. So the cell senses internal distress and sets off its own self-destruct program.
Exactly.
Okay, so it detects its own internal problems
Right.
and initiates the self-destruct sequence.
Yes.
Okay, what about the extrinsic pathway?
The extrinsic pathway.
That sounds like the signal comes from outside the cell.
Exactly, also known as the death receptor pathway.
Death receptor pathway.
It is initiated by signals from other cells,
Okay.
often immune cells,
Okay.
like natural killer cells or macrophages.
Hmm.
These immune cells produce signaling molecules
Okay.
called death ligands
Death ligands. All right.
that bind to specific receptors on the surface of the target cell,
Got it.
called death receptors.
Death receptors.
These death receptors belong to the TNF superfamily,
TNF superfamily.
which is a group of signaling molecules involved in inflammation and cell death.
Okay.
Examples include TNFR1, FES, also called CD95,
Okay.
and various trail receptors. When a death ligand binds to its death receptor,
Okay.
it leads to the formation of this complex of proteins
Uh-huh.
inside the cell membrane called the death-inducing signaling complex,
Death-inducing signaling complex, or DSC.
or DSC.
Okay.
This DSC then recruits and activates an initiator caspase,
Okay.
primarily caspase 8.
Caspase 8.
Now, depending on the specific type of cell,
Mm-hmm.
classified as type I or type 2,
Dude.
caspase 8 can either directly activate the executioner caspases,
Right.
or it might do so indirectly
Okay.
through this signaling loop involving the mitochondria.
So the mitochondria are involved again.
Yeah, they often are,
Yeah.
but the end result is the same.
Okay.
The activation of the caspase cascade and the execution of apoptosis.
It's amazing how these two pathways, one internal and one external,
Yeah.
can converge on this same controlled cell death program.
It is fascinating.
Now, I noticed the sources also talk about what happens when apoptosis goes wrong.
Right.
Too much or too little
Uh-huh.
can lead to problems.
So, the proper regulation of apoptosis is critical for health.
Yeah.
And dysregulation, too much or too little, is linked to all kinds of diseases.
Like what?
Too little apoptosis
Okay.
can lead to the accumulation of abnormal cells, which is a
Right.
key feature of cancer,
Yeah.
and can also contribute to hyperplasia,
Hyperplasia.
which is an excessive increase in the number of cells
Okay.
in organs like the lymph nodes and the liver.
Got it.
It can also play a role in autoimmune diseases,
Okay.
where those self-reactive immune cells that should be eliminated aren't. And in tumorigenesis,
And morphogenesis.
the development of tumors,
Right.
too much apoptosis
Okay.
can contribute to degenerative diseases like Alzheimer's, where there's an accelerated loss of neurons,
Right.
and inflammatory conditions like ulcerative colitis,
Okay.
where excessive cell death in the intestinal lining contributes to the disease.
So getting that delicate balance right is crucial. And this deeper understanding of apoptosis has even led to some new therapeutic approaches.
It has. Researchers are looking at ways to manipulate apoptosis for therapeutic benefit,
Okay.
particularly in cancer. For example, there's a class of drugs called BH3 mimetics,
BH3 mimetics.
such as ABT-737 and navitoclax.
Okay.
These drugs work by inhibiting anti-apoptotic proteins
Okay.
that belong to the BCL2 family.
BCL2 family.
These proteins are often overexpressed in certain cancers, especially B-cell malignancies,
Okay.
and they prevent cancer cells from undergoing apoptosis.
So they're like keeping the cancer cells alive.
Exactly.
Yeah.
By blocking these inhibitory proteins,
Okay.
the BH3 mimetics can help to restore the cancer cell's ability to undergo programmed cell death,
Got it.
effectively killing the cancerous cells.
That's a really powerful example of how understanding these fundamental cellular processes can translate into tangible medical applications.
Absolutely.
Now, we spent some time talking about necrosis and apoptosis, the two classic forms of cell death.
Right.
But the material you shared also made it clear that our understanding of how cells die has really expanded.
It has.
And it's definitely not just a binary system anymore.
That's a key takeaway.
Okay.
We've moved beyond this traditional view of apoptosis as the primary form of programmed cell death.
Right.
And necrosis as purely accidental and uncontrolled.
Oh.
We now recognize this much more diverse array of cell death types,
Got it.
which are broadly categorized into programmed cell death and inflammatory cell death.
Okay, so programmed cell death
Yeah,
and inflammatory cell death.
right.
And the review diversity and complexity of cell death really delves into some of these
One does.
more recently discovered mechanisms.
Yeah.
So can you give us a glimpse into this expanded universe of cellular demise?
Let's start with autophagic cell death,
Okay, autophagic cell death.
which is sometimes called type 2 cell death.
Okay,
Autophagy literally means self-eating.
self-eating.
And it's this fundamental process where cells degrade their own components, things like damaged organelles or misfolded proteins, by engulfing them in these double-membraned vesicles called autophagosome.
Autophagosomes.
These then fuse with lysosomes,
Okay,
which contain digestive enzymes
right.
to break down the contents.
Got it.
While autophagy is initially a survival mechanism,
Right.
allowing the cell to recycle resources during times of stress or nutrient deprivation,
Right.
it can also, under certain circumstances, lead to cell death.
Oh, so it's like a survival mechanism that can go too far.
Exactly. And the term autophagy in its modern context was largely defined by Christian de Duve in the 1960s.
Wow, back in the 60s.
Yeah.
Okay.
The process involves this complex network of proteins, including the ULK1 complex
ULK1 complex.
and various ATG proteins.
BTG proteins.
Morphologically, it's characterized by these large numbers of cytoplasmic vacuoles.
Cytoplasmic vacuoles.
And interestingly, autophagy has a bit of a dual role in cancer. It can sometimes promote cancer cell survival and resistance to therapy.
That doesn't sound good.
No, it doesn't.
Okay.
But in other contexts, it can actually contribute to cancer cell death.
Okay, so it's a double-edged sword.
Yeah, it really is.
So in some cases, the cell is essentially eating itself to death.
That's one way to put it.
That's a pretty striking image.
Yeah.
What about pyroptosis?
Pyroptosis.
The name definitely suggests something fiery.
That's a very accurate association.
Okay.
Pyroptosis is a form of programmed inflammatory cell death.
Okay. Inflammatory cell death.
And it's distinct from that non-inflammatory nature of classic apoptosis.
Okay.
It was initially observed in macrophages
Macrophages.
when they were infected with certain bacteria,
Okay.
like Salmonella and Shigella.
Got it.
The key event in pyroptosis is the activation of an enzyme called caspase-1.
Caspase 1.
And this activated caspase-1 does two things.
Okay.
First, it leads to the release of potent inflammatory cytokines,
Like
interleukin-1 beta or IL-1
IL,
and interleukin-18, IL-18,
IL-18.
into the surrounding tissue,
Okay.
which triggers inflammation.
Right.
And crucially, it triggers the formation of pores in the cell's plasma membrane.
So the membrane starts to get holes in it.
Exactly, this leakage.